For chapters 19 through 13, we're going to do it a little bit differently. I don't actually have audio from live lectures, but rather what I'm going to do is I'm going to just kind of go through the PowerPoint and point out, some of the main things that I want you to get from the PowerPoint, and then also to sort of help you to focus on the stuff that you should be looking at in the chapters in the book. So chapter 9 is about DNA and the molecular structure of chromosomes. It's a pretty straightforward chapter. What we're going to do in this chapter is we're going to look at what are some of the functions that genetic material have to perform, basically the three major functions. We're also going to look a little bit at some of the experiments historically that sort of proves that the genetic information was stored in DNA, primarily in DNA, and then we'll also, towards the end, just kind of look at the bare bones structure of DNA and RNA. We've discussed the association between genes and chromosomes, but as of yet we haven't necessarily looked at the discovery of DNA itself, nucleic acid, and then DNA itself as the carrier of genetic information. These are the three criteria that had to be satisfied. We can look at those criteria and then say, okay, what are the function of each of these, and sort of name the function, and then associate that with these different things that we've talked about. So the first criteria, to be able to replicate, that's the genotypic function. We've seen that in DNA replication. The second criteria, to control the growth and development, is a phenotypic function. This is what we call gene expression. And lastly, allowing the organisms to adapt. This is an evolutionary function that more specifically is what we call mutation. If you can't think of any, here are some hints. Chapter 5, chromosomal theory, Drosophila. So historically we kind of had two candidate materials, right, that could be the substance of this hereditary material. It could have been proteins and it could have been nucleic acids. Within the nucleic acids, we know, and just sort of as a reminder, there are two different kinds, general kinds, deoxyribonucleic acid and ribonucleic acid. So we're going to talk about some of this proof, but also note some exceptions to DNA as being where the genetic information is encoded. We'll talk at the end of the chapter about each one of these exceptions and go into it in a little bit of detail. So in the first half of the 20th century, most scientists thought that protein was the best candidate. How come? Let's think about it. If we think about what proteins are made of, what are they made of? I'll pause and let you answer. Amino acids, right? How many standard amino acids are there? 19 to 20. And if you have that many amino acids, how many different combinations of 3 can you make from those? Let's just take 20. If you repeat, you can have 20 possible amino acids in the first position, 20 in the second, 20 in the third. 20 times 20 times 20, times that up, and it's a lot. If you do that same calculation for 3 positions of base pairs, there's 4 possible ones. 4 times 4 times 4 equals 64. Much fewer combinations. So how can so many different characteristics that we see out there, physical sort of morphological characteristics in the real world, be accounted for by a code that doesn't sort of intrinsically possess as much information? And the solution is that even if you don't have very many different states, if you have a long code, you make the code the code longer, then you can account for that. We can see this similar thing going on with languages. In English, we have 26 different letters. In Hawaiian, that language only has 12. Binary code, we only have 2, but we can account for all of those different combinations. How do we account for it? If we just compare in a general sense, Hawaiian language and English language there, that is the state fish of Hawaii. And as you can see, all you do is you make the names longer, right? And so just in a general sense, that's how it's done. With the DNA code, we just make the DNA, you know, not the combinations, but the total length of the DNA, or the total length of the material that is housing that information, much longer. It wouldn't have to be so long with amino acids, but for one reason or another, even historically, it just happened to be a nucleic acid. Early indirect evidence that suggested DNA was the material we were looking for. One of the first experiments done to sort of link DNA to genes was one done by a British medical officer by the name of Frederick Griffith. He was doing these experiments in the 1920s. There was a lot of interest in vaccines at the time. They were just coming off of the end of World War I, where infectious disease caused most of the casualties. Also, the global epidemic, the global Spanish flu epidemic, had just sort of ravaged the globe, and so there was a lot of interest in this. And so he was trying to develop a vaccine for the bacterial infection, a bacterial pneumonia infection from Streptococcus pneumoniae. And he was working with a couple of different strains. One strain, type 2R, as you can see here in the illustration, if he injected that type of strain, live strain, living bacterial strain, into a mouse, nothing happened. The mouse or mice did not contract pneumonia, so the mouse lives. That was one sort of observational thing. Griffith also worked with the type 3S strain. If you took living S strains and injected them into the mice would develop pneumonia and die. If before injecting them, though, he exposed them to high heat and heat killed the S strain, he would then inject them into the mice, and the mice would live. They wouldn't contract pneumonia. And so something was going on. Let's think about this. What's going on with the heat killed strains? So the really interesting thing happened when Griffith injected a living R strain and a heat-killed S strain into the mice, mixing both of those. When you mix both of those individually, neither of them killed the mice, right? But together, the mice developed pneumonia and died. And so what's going on? I want you to think about this after you've thought about it for a little while. If you go to chapter 8, the genetics of bacteria and the viruses, and look on page 173 under the mechanisms of genetic exchange in bacteria and transformation, it'll tell you a little bit more about what's going on. But obviously something is getting passed from the heat-killed S strain into the living R strain. The living R strain has all of the material to produce new bacteria, and the heat-killed S strain also has some information, right? And so whatever heat-killed the S strain, now let's think about nucleic acid, DNA versus protein. Which one is more heat stable? So think about those things, and then go to the book and read that. So Griffith never discovered an effective vaccine, but this is really a turning point for looking at or discovering what the actual genetic material was. Later on in 1931, a pair of researchers did the same kind of experiment, but they, rather than doing it in mice, they did it in vitro. If you go to that same place in the book that I just talked about, page 173 in chapter 8, it also gives a little bit more of the details of that experiment. Here's a figure that shows the experimental design. This experiment is obviously more straightforward. You don't have to deal with the complexities of a living system like a mouse. But even with this in vitro experiment, there's still some complexity. Are you still absolutely certain? How can you be certain that it's DNA that is being transferred and not RNA or some protein that you didn't actually get out. A third experiment dealt with this scenario, and they addressed this by adding protease, RNase, and DNase, and you can see the results here. Again, all three of these experiments are in the same section that I mentioned before in Chapter 8, and then again a little bit in Chapter 9. So we've already kind of talked about previously in the semester the structure of DNA and RNA, but here let's just sort of reiterate it. DNA is double-stranded. Adenine pairs with thymine, guanine pairs with cytosine. RNA is usually single-stranded, and rather than containing thymine, it contains uracil. The three subunits that make up the nucleotide, a phosphate group, a sugar, pentose, and a cyclic nitrogen-containing base. Phosphate groups can have a single phosphate when there's that phosphodiester bond, or can have a triphosphate. Here we see the difference between RNA and DNA. No hydroxyl on a 2' carbon in DNA gives it the deoxy sense to the name. Again, just sort of as a reminder, look at the carbon atoms and see how we have labeled them: 1', 2', 3', 4', 5'. This will eventually give orientation to the DNA molecule. Here are the nitrogen-containing bases. Obviously, well, not obviously, but I don't really want you to memorize the structure for these. It's, more detail than is probably what is useful. But I do at least want you to kind of look at the general structure of them. If you look at the pyrimidines and the purines have those the two rings. The pyrimidines just have a single ring to them, and so just kind of have a look at these bases. When we put them all together, all the subunits together, this is what the nucleotides look like. When we link those nucleotides or the monomers to each other and create the polymer, what ends up resulting is a single strand of a DNA molecule, call it the polynucleotide chain. You can see that the connection between each of the nucleotides is through that phosphate group. That's that phosphodiester bond between the sugar and the phosphate. Towards the right side of the figure we see there, that's where the nitrogen-containing bases are oriented. That will be directed towards the interior of the DNA molecule. Again, look at the very top nucleotide there. That is the 5' end of the strand, because you have the 5' carbon at the very top. At the bottom, you have the 3' end, because you see there, in this case at least, it would be normally a hydroxyl, but just the way that the figure was created, it sort of shows that phosphate at the bottom connected to the 3' carbon. This is important. We'll get more into these details when we talk about replication in the next chapter. This table shows results from Chargaff's experiments, where he basically looked at the base composition of DNA in a bunch of different organisms. I want you to look through that and see what you can conclude. This is what Chargaff concluded. So now, what does that tell us about the structure of DNA? Or more specifically, what does that tell us about the association between the nitrogen-containing bases? Which bases bond with which base? The final discovery of the structure of DNA was made possible due to the research of two researchers, Rosalind and Maurice Wilkins, who in the 1940s used X-ray diffraction techniques to generate a whole bunch of data that were then sort of used by James Watson and Francis Crick to eventually come up with the double helix model of DNA. The link at the bottom of this page is a nice little 10-minute video on on that whole history, and then also on some of the history of some of the experiments that we've already talked about up until this point. Here are a number of different depictions of the molecule. Here's a table that shows some of the chemical bonds that are important for DNA structure. It'll probably be review for a lot of you, but read through those and understand them. Most of you probably already do. If you don't, come talk to me. So this is a space-filling diagram of the DNA double helix. You can see here in the middle, in the core, you have stacking forces that are at play between adjacent base pairs. This contributes significantly to the stability of the molecule. This particular form is the wet form, or the B helical form. This is the form that you see within the the aqueous environment of the cell. There are other alternative forms of DNA that the book talks about. There's the dry form, or the A form, and then there's also the Z form. And so depending on the environment that the DNA molecule finds itself in, you see that it can actually take slightly different forms. The diameter of the helix will change with each of the forms. The number of base pairs per turn with each of the forms will change. It talks a little bit more about this in the book if you want to read a little bit more about it. Here, the important thing is the opposite polarity of the two strands. You see there on the left, you see there's 5' to 3' direction on the first one there on the left, and then it's 3' to 5'. They're reversed. So that's an important thing to note. The 5' of one of the strands is associated, or on the other side of that is the 3' of the other strand, and then the 3' of the initial strand, opposite of that 5 of the opposing strand. Also note the hydrogen bonding between the base pairs. As you can see with A and T, with thymine and adenine, you have 2 hydrogen bonds, and between guanine and cysteine, you have 3 hydrogen bonds. And so there's more forces, there's more bonding forces between G and C, base pairs than between A and T base pairs. So depending on the composition, the GC competition of a strand of DNA or a particular gene, you'll see that you need to denature, or to separate the two strands. You need to expose temperature depending on the composition of G and C. So if there's more GC bonds, then you're going to have to expose it to a higher temperature. And this is important later on when we talk about polymerase chain reaction, PCR reaction. In separating, the initial step is separating those two strands. And so if the particular strand that you're amplifying has a lot of GC, you need to expose it to a higher temperature. So the last section of Chapter 9 that we're going to talk about is a discussion on other genetic material. In 1957, Heinz Frankel-Conrat did a number of experiments with the tobacco mosaic virus, where they were able to show that RNA could also serve as genetic material. This diagram shows the basics of the experiments. Basically what they would do is they would degrade each of the two types of viruses, shed them of their protein coat, expose them to the opposite protein coat, so you get this sort of hybrid type of virus, and then they would expose the virus to the tobacco leaves, and as the viruses would reproduce, it showed that the RNA actually, in subsequent generations, would produce protein coats of their original type. So at the end there you see RNA A producing protein A, therefore showing that the RNA was actually the genetic material in future generations. Another type of genetic material are viroids, which are essentially naked RNA molecules with no protein coat. They can be important in agriculture. Here is a picture of a potato spindle tuber viroid. As you can see, it's pretty small. It's only 357 nucleotides, but it can cause big problems for crops. The most interesting alternative genetic material, though, are, well, in my opinion, are prions. Prions are infectious proteins. These proteins exist in a misfolded state. This is a pathogenic state. And the misfolded proteins act as templates that convert normal proteins into the infectious protein form. So in this sense, the genetic information, this misfolding information, is then passed from one protein to the next, and these misfolded proteins then recruit more misfolded proteins. Problems arise when these misfolded proteins accumulate in the central nervous system, cause spongy degeneration, and eventually death. Mad cow disease is caused by prions. Here is a link to a video that tells you just a little bit about it. Most of you have probably heard of mad cow disease, or read about it, or even seen videos about it. The reason mad cow disease makes the news every so often is because what happens is these misfolded proteins are actually really, really stable. And so they can be passed along from cow to cow or from cow to humans simply by consuming the misfolded proteins. In the cow industry, historically, cow feed has been supplemented with protein. The protein source was other cows, and so they chop up dead cows, put it into the feed, and then feed it to the other cows. So as you can imagine, if you have a cow if you have a cattle that is testing positive for mad cow disease, and that cow was used to supplement the feed of other cattle, then you could have a massive spread of mad cow disease. Also, the incubation periods are on the order of years, and so you can have a cow eat a protein, a misfolded protein. The accumulation begins, and it may be, a long, long time, sometimes decades, until it manifests itself. And so we tend to, really kind of react in the extreme when we find cases of mad cow disease. We slaughter lots and lots and lots of cattle. Here, this is a figure from a couple of years ago. About 179,000 cattle have been affected, but about 4.4 million have been slaughtered just because, we're sort of paranoid that a cow may have mad cow disease and it just hasn't manifested any of the symptoms. A similar condition, chronic wasting disease, happens in deer populations. Same sort of thing is going on. Here, this is a picture of a deer that is suffering from chronic wasting disease. We have had cases in Utah with our mule deer and elk population. Well, elk, just 2015 was the first case of an elk, a bull elk down near Blanding at a private ranch, tested positive for chronic wasting disease. But ever since about 2002, we've had recurring reports of chronic wasting disease in our deer populations. Most of them have been in northern Utah over around Vernal, and in southern Utah in the La Salle Mountains. That happens to be the place with the highest incidence of chronic wasting disease, somewhere in the bull or in the buck population, somewhere around 2% down in the La Salle Mountains. So if you're deer hunting, those of you who like to deer hunt, and especially if you're deer hunting in the La Salle Mountains and you see a buck that looks like this picture, even though you may want to have the antlers on your walls, please refrain from shooting a deer that looks like that. This is what prion disease looks like in humans. The Creutzfeldt-Jakob disease is the most common prion disease in humans. Kuru is another human prion disease. It's best known for its epidemic levels of certain populations in Papua New Guinea, certain indigenous tribes down there. Um, practice, or at least practice until about 40, 50 years ago, the ritualistic consumption of dead relatives, cannibalism as part of their funeral rites. As you can imagine, this would pretty readily spread prion disease if you're consuming other individuals that have prion disease. In Papua New Guinea, it's also known as laughing sickness because of these pathological bursts of laughter from infected people. It's thought that it has incubation times between 30 and 50 years. This may be a little bit on the long side. Part of the estimation of these incubation times were based on the fact that the government of Papua New Guinea outlawed cannibalistic consumption 30 to 50 years ago, or something like that, and then they would have cases occurring after that, and so the assumption was that people stopped, participating in cannibalism. The obvious, flaw with that logic is possibly that even though it was outlawed, people still continued to practice cannibalism. One interesting fact with prion disease in Papua New Guinea is within the 4A tribe is that there's 9 to 10 times more prevalence in the females and children than in males. If we think about this for a second, we would say, okay, well, obviously something's going on. Two things may be happening here. It may be that males are getting the better, more, it's kind of gross to think about, but the choicer cuts of meat on the dead relatives. But more likely than that, more likely than the females having to eat the organs and the brains and, the parts of the dead relatives that maybe aren't as flavorful or desired. The other thing that may be going on is that more likely the female and children are more involved in the preparation of the bodies for consumption, and while they are preparing the bodies, they get exposed to a lot more of the tissue of the deceased person. So again, review the key points. And that does it for Chapter 9.